technologies. How many of you are familiar with this or have heard about it? Just tiny little bit? Okay. So the first half of the lecture we will talk about copy number variants, and then in the second half we will cover chromosomal microarray technologies which are used to detect these copy number variants. So as you all know, humans have 46 chromosomes. 23 from mom, 23 from dad. And we call these homologous chromosomes. So 2 chromosome number 1, 2 number 2, and so on. So each copy or each chromosome has 2 copies. So theoretically speaking, any piece of DNA or any stretch of DNA should have 2 copies, right? That's the normal. One on the maternal homolog, one on the paternal homolog. Any deviation from this is considered to be a copy number variant. So these are stretches of genomic DNA which are present in more than or less than 2 copies, and they usually range in size from kilobase to megabases. So they are pretty big chunks of DNA. Unlike the base pair changes, which can be 1, 2, or few base pairs, these are big structural changes in the genome. So again in the copy number, which we will call a triplication, duplication. One copy number gain will be a duplication, a 2 copy number gain will be a triplication, a 1 copy loss is a heterozygous deletion, a 2 copy loss is a homozygous deletion. So these are changes in the copy number. As I said, they can range from the kilobase to the megabase range. They cannot be identified by more conventional cytogenetic techniques like karyotyping, because karyotyping has a resolution of approximately 5 megabases. It cannot see anything less than 5 megabases. So it can only be detected by chromosomal microarray technologies, which we will discuss in the second half of this lecture, and nowadays with whole genome sequencing. Whole exome sequencing is can be challenging to detect copy number variants, but whole genome sequencing, if you use the right algorithms and you do enough sequencing depth, you can detect these CNVs. These CNVs, that's the short term for copy number variants. They can be germline, they can be somatic, so they can be present in all body cells, so they are in the germline, or they can be somatic, present in some cells and absent in others. They can also be sporadic, which means that they are de novo, or they can be inherited. And usually the large de novo CNVs, these can be disease causing. Recent studies have suggested or showed that CNVs are widespread in the human genome, and they are a significant source of human genetic variation, accounting for population diversity and human disease. And they are quite common in the human genome. So if you add up the base pairs contained within these CNVs, they will be more than 100-fold higher than what's present in SNPs. Their phenotypic effect can sometimes be unclear, and it all depends whether they span what we call dosage-sensitive genes. Dosage-sensitive genes are genes that cannot be tolerated if present in less or more than 2 copies. They have to be always present in 2 copies. If present in 1 copy or present in 3 copies or more, they can give a phenotype. And in a clinical setting, CNVs, when we do this as a clinical diagnostic test, we have to categorize these CNVs into one of those 5 categories according to the American College of Medical Genetics guidelines. So they can go all the way from benign to pathogenic. These are the extreme ends. And then everything in between from likely benign, likely pathogenic, and a variant of uncertain significance. And there are rules or there are guides on how to make these interpretations. Now, this figure shows you the size and the frequency of genetic variation in general in the human genome. And it's a very simple figure, but it summarizes what kind of variation you can see in the human genome and how frequent it is. So on one end, you have the base pair changes, like SNPs. These are very common and they are very small in size. Their effect can be big, they can be small on the phenotype. On the other end, you have big chromosomal rearrangements, a whole chromosome loss or a whole chromosome gain, or a whole chromosome arm loss or a whole chromosome arm gain. These are very rare, or not very rare, but they are relatively rare, but their effect on the phenotype is so drastic, giving genetic disease. And between this end and that end lies the copy number variance. Believe it or not, until approximately 10 years ago, there were techniques to see this end, which is karyotyping. There were techniques to see this end, which is SNP genotyping. But we were completely blind to what's in the middle because the techniques available to see these CNVs didn't exist until the introduction of chromosomal microarrays. So this is another important table that shows you the difference between genomic rearrangements, including CNVs, and base pair alterations, which are the single nucleotide changes. So size-wise, these CNVs, are in the KB to the megabase range, whereas the base pair alterations, they are quite small, like point mutations. As I said, 1, 2, or few base pairs. The gene content, this is only one gene at a time, whereas this can contain or span several genes. The molecular mechanisms resulting in these are very different. I will not go into them into huge details. Another important difference is the mutation rate. The mutation rate for CNVs is actually higher than SNPs. And the methods for detecting them is very different. G-banded chromosome analysis or karyotyping in fish, this can only see things, as I said earlier, bigger than 5 megabases. Fish or fluorescence in situ hybridization is a very useful technique, but you can only use 1 or 2 probes at a time. And chromosomal microarray, which we will discuss in more details in the second half of the lecture, which can actually cover the entire human genome looking for these CNVs. On the other hand, for base pair alterations, you can use sequencing or other molecular techniques. I'm not really sure, but it has to do with the architecture of the genome and how it's prone to these kind of structure rearrangements during meiotic division. So now, not all CNVs are pathogenic or bad. The majority actually of CNVs in our human genome are what we call benign CNVs. So a recent estimate for the proportion of the human genome which is structurally variant by benign CNVs estimates that it's between 5 and 10%. So if you take 2 individuals and do a in-depth CNV analysis, you can see that up to 5 to 10% of the genome can be different between 2 individuals. The majority of these benign CNVs, more than 95% of them, are smaller than 100 kb. So they are quite small. The ones that are bigger than 100, especially the ones that are bigger than 500 kb, they tend to be more pathogenic. And there are online tools or databases to look for these benign CNVs. And one of them is the database of genomic variants, which actually is a catalog for benign CNVs that were observed in healthy controls from many studies. And this snapshot from the UCSC genome browser, which we will discuss in the second lecture, my second lecture, shows you a track here for the database of genomic variants. So just if you're not familiar with the UCSC genome browser, have you ever played with it? Any of you? So the UCSC Genome Browser is a tool that will allow you to zoom in into the human genome and look what's there. Genes, regulatory elements, many other things. So I'm showing here few things that are relevant to this talk. So we are zooming on a stretch of chromosome 22, and you can see here a genomic ruler. So in base pairs, how many base pairs? You have the cytogenetic bands, you have the reference sequence genes, and you have here at the bottom something called segmental duplications. These are structural elements in the human genome that make it sometimes prone to these kind of structure rearrangements. And this track here that has the blue and the red bars are the benign CNVs in the human genome. The blue ones are duplications and the red ones are deletions. So, and these are actually from different individuals. So you can see that, for example, here you have a cluster of benign CNVs and some of them contain genes. So within these CNVs, one person can have 2 copies of a particular gene, another person can have 3 or 4 copies of that gene, and a third person can have just 1 copy of that gene. So some of them contain genes. And if you look closely, many of them actually overlap these segmental duplications. Questions so far? So can CNVs cause disease? Yes, they can. Most of the CNVs, as I mentioned earlier, are benign, majority of them. But some of these benign CNVs actually can encompass genes, as I showed you, especially the genes of the immune and environmental response pathway. And this suggests that they have an important role in local adaptive selection in human population. So these are kind of normal variations between human beings. But some CNVs, especially the big ones, and the ones that contain developmental genes, like for example, a transcription factor necessary for development, these guys can result in human genetic disease. Some CNVs, some of the pathogenic CNVs actually encompass several genes. And each one of them, especially the ones that are dosage sensitive, they can contribute to the phenotype independently. So your end result, your end phenotype is kind of the additive effect of the extra copies or the less copies of these dosage sensitive genes. And clinically, when we do this kind of chromosomal microarray analysis clinically for patients, we see that approximately 18 to 20% of the patients have these pathogenic CNVs. So the diagnostic yield of the technology is up to 20%. That's why in 2010, the American College of Medical Genetics suggested that these technologies, the chromosomal microarray technologies, be the first tier test for individuals who have developmental delay, intellectual disability, other neurodevelopmental issues like psychiatric issues, as well as patients with multiple congenital anomalies. So how the CNVs cause disease? The first mechanism, as I showed you earlier, is if they span a dosage-sensitive gene. And a dosage-sensitive gene is a gene which will give you a phenotype when present in more than 2 copies or less than 2 copies. Another mechanism is gene interruption. If you have a break in a gene, so if it's a deletion, it will have the same effect like the gene dosage. But how about if it's a duplication? So if it's a duplication and the duplication is oriented in a certain way, if it's an inverted duplication, it has the potential to disrupt the reading frame of the gene. And instead of causing an increase in the number of copies of the gene, it can result in a decrease in the number of copies because one of the copies is interrupted by the breakpoint and you actually lose this copy. Gene fusion. So if you have a break in gene A another break in gene B, and you fuse these two genes in tandem. This is more common in cancer, where you fuse the 5' end of one gene with the 3' end of another gene, and you result in and they are fused in frame. This will give you a fusion gene which has new properties, and in this case, oncogenic properties. It can drive the oncogenic process. Position effect. So how about if the CNV doesn't really span the gene, it spans the regulatory elements that control that gene. So it can, let's say that this is a deletion, and it spans the enhancer elements that control this gene. Deleting the enhancer elements will down-regulate the gene as if you have deleted the gene. Also, unmasking a recessive allele or a functional polymorphism. So imagine if you have a point mutation in one gene, and then you delete the other copy of the gene by a CNV, and this is a recessive gene, so you actually knocked both copies of the gene. This will give you a phenotype. So these are some of the mechanisms how CNVs can give a phenotype. Questions? Good so far? This is a table that was published in 2010 that help us interpret the clinical significance of CNVs in a clinical setting. And you don't have to go every point here, but these are just clues or guidelines that help us determine the pathogenicity of a CNV. For example, if it's de novo or inherited, if it spans known dose-sensitive genes, if it overlaps with benign CNVs or not, if it was inherited from a parent who has a similar phenotype, and so on. So, I mean, these are all things that we use to determine the clinical significance of these CNVs. CNVs can be categorized into two groups: recurrent and non-recurrent. Recurrent CNVs are those that have clustered breakpoints. So if you take a patient or an individual with a certain deletion, and you compare the breakpoints of the deletion with another person who has a similar deletion, you will find that the breakpoints of the two deletions are very, very similar. They are clustered in what's called the flanking segmental duplications. So these segmental duplications are the architectural elements in the human genome that will mediate these recurrent deletions or duplications. There is a specific mechanism for that called non-allelic homologous recombination. I will not go over it in huge details, but if you are interested, you can talk to me after the lecture. But these flanking segmental duplications will mediate CNVs, deletions or duplications between them, and therefore the breakpoints of the deletion or the duplication are always clustered in these flanking segmental duplications. That's why we call them recurrent. Another type of CNVs are the non-recurrent, where the breakpoints are all over the place, but they will overlap. If you take one CNV, compare it with another from another individual, compare it with a third from a third individual, and so on, they will overlap with what we call the smallest region of overlap, which usually is the region that contain the gene or genes that give you the phenotype. So that's why, or that's how these patients are ascertained. But the breakpoints are all over the place. So what are the mechanisms that generate these CNVs? Non-allelic homologous recombination is the mechanism that generates recurrent CNVs. Two other mechanisms, non-homologous end joining and fork stalling and template switching. These are two mechanisms that mediate the non-recurrent CNVs, the ones that don't have the clustered breakpoints. The breakpoints are all over the place. Questions so far? You will hear other terms in the literature, microdeletions and microduplications. Microdeletions and microduplications are practically CNVs. The reason they call them micro is that they are smaller than the resolution of karyotyping. So they are smaller than 5 megabases. They are practically speaking CNVs, but they are CNVs that produce phenotypes. They are pathogenic CNVs that produce phenotypes. Many of them have been characterized clinically before the genetic basis was known. So for example, if you've heard of the DeGeorge syndrome, Williams syndrome, Prader-Willi-Angelman syndrome, these are all clinical syndromes that are due to microdeletions that were described clinically based on the phenotype before their genetic basis was known. Later on, it was shown that they are due to microdeletions, small deletions that cannot be picked by karyotyping. You need more molecular techniques like chromosomal microarray or FISH if you know what to FISH. Some of them, especially deletions, microdeletions, if they are on the X chromosome in a male, you are actually deleting the only copy of that gene in a male because the other copy is a Y chromosome. And you are resulting in structural nalosomy. You are deleting the single copy available in this male. And if they encompass imprinted genes, are you familiar with that term, imprinted genes? So imprinted genes are genes whose expression vary on the sex of the patient or the individual. So if it can be imprinted or silenced in a male and active in a female or vice versa. So if you have a deletion or a duplication that spans imprinted genes, you will find that its phenotype can be different depending on whether the deletion or duplication is on the paternal or the maternal homologue of the chromosome. So the imprinted genes, their expression vary whether on it's a paternal or a maternal homologue of the chromosome. So for example, the same deletion on chromosome 15 can produce either Prader-Willi syndrome or Angelman syndrome. If it's on the paternal homologue of chromosome 15, it will produce Prader-Willi syndrome. If it's on the maternal copy of chromosome 15, it will produce Angelman syndrome because the genes that are within this region, their expression varies depending on the chromosome, whether it's paternal or maternal. So if you hear this term, microdeletions or microduplications, these are clinically significant CNVs. This is just a different naming. They are smaller than the resolution of karyotyping. They are smaller than 5 megabases. And technically speaking, they are CNVs. Yes?
If you have a de novo mutation, how do you link genotype to phenotype? If you're not sure what the gene is that underlies intellectual disability.
So if the deletion contains genes that have been characterized before, you can infer the phenotype or kind of make a correlation based on the gene function. If it has been shown before in other patients to give a phenotype, that would be even more helpful. I'll go into this more in the second lecture, and I'll show you the UCSC genome browser and how to use it and look at the gene content and how to look into each individual gene and how to use the UCSC genome browser to link out to other databases to get more clues. So it all depends on the gene content and what has been known about that gene, whether from research studies or from other clinical studies. So there are online tools. So I mentioned earlier the database of genomic variants, which is a catalog for benign CNVs. There is a similar database which is clinically based, which is the Decipher database. This is the database of chromosome imbalance and phenotype in humans using the Ensemble resource. This is a catalog of actually pathogenic CNVs, especially the recurrent microdeletions and microduplications. They are shown here. There have been 70 of them described in the literature so far. These are microdeletions and microduplications that are recurrent and have a relatively consistent phenotype. Now, in the literature, research studies using or CNV research studies over the last 5 years has focused on patients with neurodevelopmental issues. And it has been shown that patients with neurodevelopmental issues have a significant burden of CNVs, more than the normal individuals. And the burden of CNVs correlates with the severity of the phenotype. So these are two graphs taken from two studies that shows you the size of the CNVs and the frequency of the CNVs. And as you can see, the more severe the phenotype, the more and the bigger are the CNVs. So patients with craniofacial abnormalities, shown here in the blue in the red graph, patients with intellectual disability, patients with epilepsy and autism. So autism would be the smallest burden, and craniofacial abnormalities would be the highest burden of these CNVs. So the more severe the phenotype, the more the CNVs in number, and the bigger they are, so they will span more genes. And it has been shown that many of these genes, especially in neurodevelopmental issues, they are genes that have neurodevelopmental functions. So they are either transcription factors involved in the development of the brain or the central nervous system, or they are adhesion molecules that function in the synapses, which are the junctions between the neurons. And many of those genes have been shown to be encompassed within or included within these CNVs. Also, genes that encode neurotransmitters have been shown to be involved in these CNVs. So again, this is just to show you that these things are quite common, both in a pathogenic form and in a benign form. Also, these CNVs can have variable expressivity. So this is a list of some of the recurrent CNVs or microdeletions or microduplications, and they are named after the chromosome band. So for example, here this is chromosome 16, P11.2. 16 is the chromosome, P is the short arm, 11 is the region, the band, and 0.2 is the subband. So this is a deletion or a duplication in this region of chromosome 16. It's called 16p11.2. This would be 15q13.3. So some of these microdeletions, they don't have names, like for example the George microdeletion or Prader-Willi or Williams. They are named after the band where they sit. Many of these guys don't have a very consistent phenotype. They can present with a variety of phenotypes, from more severe to less severe, and it all depends on other hits in the genome or other CNVs in the genome. So that's what we call variable expressivity for these CNVs. So any questions before we go into the second half of the lecture, chromosomal microarray technologies? So now you are familiar with the term CNVs. They can all be present in benign form, in a pathogenic form, and so on. Yes.
So, like, you have like a patient with neurodevelopmental disorders, and you can see that they have like a greater number of CNVs, larger CNVs. Do you see that like grow over generations? Like, if you were to compare it to their parents or grandparents, or
I'm not sure if this has been studied in a multi-generation kind of thing, but what has been shown is you will have, for example, two parents who have some sort of mild neurodevelopmental issues. They have, for example, one of them has ADHD, and another one has epilepsy. But they will have a child who has a much more severe form of neurodevelopmental issues, like intellectual disability. And what's thought to be that CNV is inherited from mom plus the CNV is inherited from dad, working along the same neurodevelopmental pathway, will knock down enough genes to make the phenotype much more severe. So that's what has been shown. Plus, it's not always CNVs. You can have the second hit or the third hit in the form of a point mutation in a gene or a functional polymorphism in an important gene. So it's a combination of CNVs as well as base pair alterations. So, other questions? So let's move on to the technology part of the lecture. How can we actually detect these things? So the general term is called chromosomal microarray. And the chromosomal microarray technology was introduced approximately 10 years ago and was very important in detecting these CNVs. Where they are, where are the hotspots, where are the benign CNVs, where are the pathogenic CNVs, and so on. The original technology, so the general term is called chromosomal microarray. Under this term, there are two distinct technologies. One of them is called array comparative genomic hybridization. And this is the original technology that was introduced approximately 10 years ago. And it's very, very useful in detecting these CNVs. Another technology is using SNP arrays. Mike probably has talked about SNP arrays for genotyping. SNP arrays were introduced also approximately 10 years ago or more, but they were introduced not for CNV analysis, they were introduced to do SNP genotyping for GWAS studies and other research studies. But the companies that manufacture SNP arrays were able to find an algorithm to use them for CNV analysis. And we will get into that. So this is the original technology, array CGH or array comparative genomic hybridization. It all depends on labeling your patient DNA with one fluorophore, and in this case it's red, a fluorescent dye which is red, your control DNA with another fluorescent dye which is green. You mix these two guys together in equal quantities and hybridize them to an array. Now the array will contain thousands of spots. Each spot is an oligo representing a piece of the human genome. And within the borders of these spots, you have hundreds of these oligos. But the oligo sequence is the same. So what will happen is your patient and your control DNA will compete with each other for the target. And if they are present in equal amounts within that region, they will hybridize equally to that particular spot. Does this make sense? So let's say you have 100 oligos, all the same sequence within a certain spot. So if your patient contains 2 copies and your control contains 2 copies of a particular piece of DNA, you will find that 50% of the oligos within that spot will hybridize to the patient, the other 50% will hybridize to the control. And the end result is a ratio of red to green, which is equal to 2 to 2, basically 1. And 1 here is represented by the yellow. So the ratio of red to green dye is what we measure, and it's represented later on a graph. So after they are hybridized, and remember this is done across the entire human genome using hundreds or thousands of oligos, each representing a piece of the human genome. After the array is hybridized, it's washed and scanned, and the scan will look something like this. And then using special software, the data are extracted from this scan and plotted. The way it's plotted, each dot here or each spot represents an oligo, which represents a piece of the human genome, and it's plotted like a dot along the chromosome. The chromosomes, as I'll show you in some later plots, are usually shown from top to bottom. So each chromosome has a short arm and then a long arm, and the nucleotides, the DNA level, are labeled on that chromosome from the top to bottom. So nucleotide number 1 will be at the very top, at the tip of the short arm, and nucleotide 155 millions would be at the very bottom of the chromosome. So the nucleotides are labeled from top to bottom, and the oligos also are plotted from top to bottom depending on where they fit in the human genome. Oligos that have equal red to green dye will be in the middle, will plot at the middle. This is a log2 scale, so they will have a log2 ratio of 0. So these are pieces of the DNA that are present in 2 copies in the patient. Remember, this control is a patient or a group of, not a patient, is a normal individual or a pool of normal individuals which have been studied before. So it's everything is actually compared to your control. Now, if you have a deletion, let's say, sorry, if you have a duplication, so you have 3 copies in the patient, only 2 copies in the control. So more of the red dye will hybridize to the oligos or the spots that are contained within that deletion. Let's say this deletion, this duplication. Let's say this duplication contains 10 oligos. So these 10 oligos will have more red dye. And therefore, the red to green ratio will be more than 1. In a log2 scale, it will be 0.6. The exact opposite for deletions. If you have a deletion here, a heterozygous deletion, so the ratio will be 1 to 2. Okay, so when it's hybridized, there will be more of the control dye within the borders of that deletion, and therefore the curve will shift towards the left side, and you will have a shift to approximately -1 on a log2 scale. So deletions will shift to the right, sorry, to the left, duplications will shift to the right. And when we are doing this analysis, we cannot depend on single oligos to make calls. We have to have at least 3 sequential oligos that are deleted or duplicated, gained or lost to make a call. Any questions? So there are special software to analyze this data. And I'll show you some more pictures later on. So this was the original technology that was introduced. Yes?
So you have to do like PCR before this to get multiple copies?
No, you actually extract your genomic DNA from the individual. You shear it by enzymes, not shear it, but you cut it by restriction enzymes into pieces. And then you label it using random priming with fluorescent dye. And you do the same thing with the control. And then you take your labeled DNA from the patient and control, mix them together. So there is no PCR involved. And then it's hybridized to the array.
How long did you take to PCR?
The technique takes approximately 3 days because you digest your DNA, you label it, and this takes approximately 1 day. You hybridize it to the array for approximately 40 hours. You then wash it, scan it, and analyze it. So to get the plots takes approximately 2.5 to 3 days. But to analyze the data it takes longer because you have to take the CNVs and analyze them using different tools to see whether they are benign or pathogenic or in between. So the technology itself can take between 2 to 3 days. But analyzing the data takes longer.
Okay, so this may be a very silly question. Where do you get the control DNA? Like, is it from like family member? Or, like...
The control DNA is usually one of 2 things. Either a pool of normal people, DNA from normal people, Normal meaning that they have been examined and don't have, actually it's a very good question. They don't have any issues, neurodevelopmental issues or any other issues, or it can be just one normal individual who also has been submitted to the same kind of. Now, it's important to determine why you are running the test. Because if you are running the test to look for, in a clinical setting, to look for pathogenic CNVs, this can be slightly different from whether you are running the test in a research study to look, for example, predisposition for, let's say, hypertension or diabetes. And in this case, your control has to be really studied really well to make sure that they don't have family history of diabetes, they are not pre-diabetic, and so on. And also they are within a certain age range. So it's important that the choice of the control is important, given why you are doing the technology or why you're running it. So, but it can be just one normal control, it can be a pool of normal controls. So array CGH is the original technology that was described. I mentioned SNP arrays. So these are the two technologies that fall under this umbrella, which is chromosomal microarray. Sometimes they call them cytogenomic arrays. So cytogenomic arrays, chromosomal microarrays are all the same term, and they encompass these two technologies, array CGH and SNP arrays. So we explained array CGH. SNP arrays work in a slightly different way. SNP arrays contain sequences that encompass the two alleles of a SNP. So I'm sure that you are familiar with what's a SNP, single nucleotide polymorphism. Single nucleotide polymorphism is a single base pair change and usually present in two alleles, what we call allele A and B. So these two versions of a SNP are printed on the array. And there is no control. Only the patient is hybridized to the array. And depending on the fluorescence intensity, the data are compared with in silico controls, so controls that have been run on the array historically. And using computer software and analysis algorithms, you can actually see these CNVs based on the signal intensity without having an built-in control. It's a little bit challenging compared to the array CGH. SNP arrays have an advantage in that they are actually genotyping the SNPs. So you can look also at something that's very important clinically called regions of homozygosity. These are stretches of DNA that are bigger than 3 or 5 megabases that are present in two copies, but they are completely homozygous. Why is this important? Because it helps us to see consanguinity. If the patient is result of a consanguineous relationship, we can see that. We can also detect the degree of consanguinity. That's important. And we can also detect something called uniparental disomy, or a copy of a chromosome, both copies of a chromosome coming from one parent. And that can be clinically significant without going into further details. The original array CGH technology did not have this advantage. It was superior in SNP and the CNV analysis to compared with SNP arrays, but did not have the advantage of detecting regions of homozygosity. Only recently, SNPs were introduced into the array CGH technology, and now we can detect these regions of homozygosity using what we call array CGH plus SNP arrays. So how do these arrays look like? This is what we use in the lab. It's an array from Agilent. It's called the 4x1 ATK. It actually contains 4 subarrays. Each one of them contains 180,000 probes. 120,000 probes for CNVs and 60,000 probes for SNPs. and the average spacing between the probes is approximately 25 kb within the backbone. And I'll explain what's the backbone in the second lecture. And you can actually hybridize 4 different patients on one array. This is an image of a scanned array. So as you can see, it's very dense. These are the 4 subarrays. We have different patients hybridized to each one of them. And each one of those guys contains 180,000 probes. And this is a zoom on one of them. And the software will take this image. It will identify each individual spot. If I zoom more on this, you will see these as individual spots, and will calculate the ratio of red to green dye in each spot to produce these graphs. Questions so far? Okay. So this is, again, a snapshot from the UCSC genome browser, and it's zooming on that region that I showed you earlier, the 16p11.2 region. These are deletions and duplications from different patients. So the blue are duplications, the red are deletions. And you can see that they are clustered, and the breakpoints are very similar. If there is some variation, it's minor variation. And this is from the Decipher database. So these are all different deletions or duplications from different patients. And this deletion or duplication is caused by that mechanism I mentioned earlier, the non-allelic homologous recombination. And the breakpoints are clustered in the flanking CNVs. And the flanking, segmental duplications. So these are the segmental duplications that mediate these deletions and duplications. And you can see the genes that are within that region. Now, this is a recurrent microdeletion or microduplication. You will see the deletion and the duplication in different individuals. You won't see the deletions and duplications in the same individual, of course. So some patients will have deletions, some patients will have duplications. How does it look on the array? This is how it looks on the array. So as I told you earlier, each chromosome is plotted this way. So this is chromosome 16, this is the short arm, this is the long arm, and this is the centromere. So, and each chromosome is numbered. The base pairs are numbered from top to bottom. So if you are counting the base pairs, they go from top to bottom. Each dot here on the plot represents one of the oligos on the array. So you can clearly see here there is a region where there are multiple oligos that are shifted towards the left side. This is that deletion, the 16p11.2 microdeletion, and this is a zoom on the microdeletion. When we are doing this clinically, we always like to confirm this, not really that we don't trust the technology, but to just to visualize the results using a different technology. We confirm it by FISH, or fluorescence in situ hybridization, where we use a probe within that region, the deleted region, and we do metaphase FISH analysis. So if you look closely, you will see that this chromosome 16 has 2 probes lighting up, a red and a green. So there is a red signal and there is a green signal, whereas this chromosome 16 has only the green signal, the red is missing. The green signal is a control probe on the chromosome, and the red signal is the probe that we intentionally use within that deleted region. So this confirms that this patient has a deletion of this in this region. Okay. Another reason we do this FISH analysis is if we want to study parents, it's a lot cheaper to run FISH on the parents than run an array CGH. The array is expensive, the FISH is very cheap. So if we have a probe within the region of interest, FISH probe within the region of interest, we can run either metaphase or interphase FISH analysis on the parents to see if this deletion or duplication is de novo or inherited. All this or all this technology can be, I've shown you more in a clinical setting, but this is very, very useful in a research setting. If you want to do any sort of genome-wide CNV analysis in the patient, in your research study. The same mechanism that produces deletions can also produce duplications. So here is the same thing, but in this case, the oligos here are not shifted towards the left side, they are shifted towards the right side. This is the reciprocal duplication of that same region, and this is a zoom on that duplication. This is also clinically significant. It's the 16p11.2 microduplication, and you can also confirm it by FISH. When we do FISH confirmation for such a small duplication, remember the duplication is in tandem. So the duplicated region is on the same chromosome in tandem. When we do it on metaphase chromosomes, so these are the individual chromosomes, the signals are so close together that they overlap. But if you do it on interphase cells, when the DNA is more loose and more relaxed, you will see actually the signals kind of separating. This is a list of databases, CNV databases, that are very helpful. We will go into more details in the second lecture in the UCSC Genome Browser, but these are very, very helpful databases if you are interested further in this. Now, everything I've shown you so far is mainly constitutionally oriented. So if you are interested in studying the constitutional buildup of the individual, but the same technology can be used for studying CNVs in cancer. So cancer genomes or cancer cells have tons of these copy number variants. They are acquired. They are somatic. They are only in the cancer cells. Now, why do we want to do this in the cancer? Because some of these are very important clinically or also important for research. So normally in cancer, from the cytogenetic point, we do karyotyping of cancer cells, like for example in liquid tumors, leukemias, lymphomas, and solid tumors, and we do targeted FISH. And we want to look for certain abnormalities that have clinical significance, either diagnostic, prognostic, or therapeutic, and there are many examples for this. Each one of those technologies, karyotyping and FISH, has its limitations. Karyotyping will show you it's kind of a low resolution whole genome view because you're looking at all the chromosomes but at low resolution. But you will miss small things. Whereas FISH is very useful, but it will only detect the target which you are fishing. You cannot use FISH probes to target the entire human genome. So it's not possible to FISH the entire human genome. So that's where chromosomal microarray technologies will fit in to actually cover that gap between karyotyping and FISH, and also cover the gap which is not covered by sequencing. So chromosomal microarray, including array CGH and SNP arrays, have been used extensively in the last few years, both clinically and in research, to study cancer genomes. They are very important in detecting or characterizing the genome of those cancer cells. And they are very important in looking for clinically significant copy number changes or copy number variants in these genomes. So it can be quite crazy how these genomes look like in the cancer cells. So here is an example of one patient. That's what I'm going to close with. Patient that has B precursor ALL or acute lymphoblastic leukemia. So here is chromosome 7 that shows a deletion. It's basically a single gene deletion for a very clinically significant gene. It's called the ICROS gene. It's a very poor prognostic sign. Chromosome 9, where you have a whole chromosome arm loss. And within the whole chromosome arm loss, there is a homozygous deletion it's nested within the whole chromosome arm deletion for two clinically significant genes, tumor suppressor genes, CDKN2A and 2B. There is also a distal duplication in the long arm of chromosome 9. Chromosome 12 has a loss for 12p or the short arm and has alternating deletions and duplications, as you can see here. And this is a signature for what we call chromotrepsis. Have you heard that term before? It's when the genome kind of shatters and then rejoins haphazardly. And when that happens, you get this kind of alternating losses and gains. There is no other technology other than chromosomal microarray where you can actually visualize this in cancer genomes. It can happen also constitutionally. Chromosome 13 with a deletion in the RB1 gene, which is a very clinically significant tumor suppressor genes. So the technology is very useful for both constitutional studies as well as somatic studies, especially in cancers. This doesn't mean that it doesn't have limitations. The biggest limitation of chromosomal microarrays is that it cannot detect balanced rearrangements. If you have a balanced translocation or a balanced inversion, so basically exchange within the genome that does not result in any loss or gain, you will not see that by chromosomal microarray. That's the biggest disadvantage of this technology. Also, it cannot detect low-level mosaicism. If your cells that have the CNV are less than approximately 10%, you will not be able to see it by this technology. It also depends on the size of the gain or the loss. If it's a whole chromosome arm or a whole chromosome loss or gain, you can go down to maybe 4 or 5%. But if it's a small deletion or duplication, you cannot see it if it's present in less than 10 to 15% of the cells. So these are the two biggest limitations of the technology. But other than that, it's very, very useful technology. In a constitutional setting, it's very useful clinically to diagnose approximately 20% of the patients who have these clinically significant CNVs, patients with developmental delay, intellectual disability, other neurodevelopmental issues, even multiple congenital anomalies. So, questions?